Hello team, and welcome to your next lesson on signal transduction. I've decided to make this as a YouTube so that you can watch it as you wish and work through the activity in your own time. If you choose to do this after doing some biozones, then that's totally fine. If you would like to jump straight in, then help yourself. I'm going to start at the top of, so you need the cellular signals book. We're on page 16. We're just going to look at the study design. In Vikar's words, it says that you need to know the difference in signal transduction for hydrophilic and for hydrophobic signals. And you need to know the difference in terms of the position of the receptor, whether that is in the membrane or in the cytosol, and the initiation of transduction. Now it's very clear that you don't need to be able to name or describe specific chemicals, second messengers, G protein pathways, reaction mechanisms, or cascade reactions. They are not required, okay? So what you need is a conceptual understanding. You need to be able to describe how signal transduction is initiated by these two types of signaling molecules. So what I'll ask you to do is open the PowerPoint that you've downloaded from LMS or from the Teams files. This is called Signal Transduction Animation Reshuffled. Now I need to tell you that this is not my PowerPoint. I didn't make this. A very kind teacher of biology has made it and given it to us, and we quite like it. I think it's quite a nice activity. So if you remember the cut and paste activity we did with transcription and translation, this is a little bit like that, but no cutting and sticking. You'll just be dragging sentences around inside the PowerPoint slide to describe the two different scenarios. So here you can see the first scenario is where cortisol moves straight through the membrane and joins with the receptor inside the cytosol. Now, that should tell you something immediately. How is it possible that a signaling molecule can get straight through the plasma membrane? That must be a non-polar hydrophobic molecule, okay, lipid soluble, not water soluble. So it's zoomed on through and it's bound with its receptor inside the cytosol and forms what we call a signal receptor complex or a ligand receptor complex. Now that complex stays together and can enter the nucleus in this situation. And together what it will do is actually act as a transcription factor, allowing RNA polymerase, everybody's favorite enzyme, to read the DNA and transcribe that and build messenger RNA. And the messenger RNA will then be processed as usual and leave the nucleus, find the ribosome, and translation will occur, forming the effector proteins. In this situation, they can synthesize glucose from amino acids and glycerol. And here is your sentence sort. So to summarize what we've just looked at, go through that slide again as many times as you want to, the animation, and then cut and paste these sentences into the right order. And see if you can get it to make sense. Again, you don't need to know exactly what's happening in this scenario. We're not going to test you on the exact signal transduction pathway that occurs when cortisol enters a cell. However, you do need to be able to be given a scenario like that and identify the information as shown here. So we've got the stimulus. What is the primary stimulus here? Where is the receptor located in the cell? Is it on the membrane or is it inside the cytosol? Can you describe what happens next? So the point between reception and the cellular effect or cellular response is always the signal transduction part, that intermediate part. So use this part in your notebook, this section, to practice describing signal transduction and give me the cellular response in just a few words. The second scenario, we use adrenaline, not cortisol. And you can see a difference here in the position of the receptor. So this receptor is receiving the signal on the outside of the cell membrane, so in the extracellular environment. And it's what we call a G protein coupled receptor. Now what you can see here is that as that binds, a change in conformation occurs in the proteins associated with that receptor. So we have a change from GDP to GTP, which you don't need to remember all about. And then we have the alpha subunit has broken off and joined another molecule, adenyl cyclase. And we've got a series of steps here, which you can see occurring, which make up your signal transduction. So eventually we end up activating phosphorylating protein kinase A, adding phosphate to that, and that then goes on and actually can initiate, is responsible for the intracellular changes that occur in response to adrenaline. Okay, so you don't need any of this stuff except for the fact that if you're given this as a series of events, know that that's the signal transduction. We've got reception occurring here. Where is it occurring? Outside the cell. Why is that happening? Because adrenaline is clearly a polar molecule, a water-soluble polar molecule that can't diffuse directly through the plasma membrane. And that causes a conformational change in the G protein coupled receptor that then knocks onto a cascade of many different events and finishes up with a phosphorylation of protein kinases, which cause the cellular response to adrenaline. This inhibits glycogen synthesis, so therefore releasing more glucose for the cell to use. So we know that adrenaline is something used in the fight or flight response, and access to glucose will be really important in that. So have a look through these sentences here. There's a lot of terminology. Please don't let that intimidate you. You might be faced with lots of terminology when you're in an exam, and what you need to be able to do is wade through that and try and find the key messages. Try and apply your understanding. Where's the reception part of this? Which steps involved in the signal transduction and which relate to the cellular response? Once you've done that, try and give me some description of the receptor location, the second messenger, and then what the cellular response is. If you can identify that from the sentence or on the previous page, that would be excellent. And then I think you've addressed the key dot point that we're trying to cover today. Then return to your biozones, page 66 to 69. Do not go on to page 70. What we'll do is we'll review that next week. If you have any questions, obviously post them in the chat, give me a quick buzz, or watch the video again. Well done.